Hi, everyone. Welcome to the AOCS pitch competition for the Protein and Co-Products Division. I'm Rebecca Gennard, the Director of Publications for AOCS, and I'm also the Editor-in-Chief for Inform Magazine. I'd like to start out by congratulating our ePoster finalists who've been chosen by a panel of judges as today's pitch presenters. Great job to you. That's a pretty competitive group that you made it through to the final round. And before I introduce our presenters in just a moment, I would first like to acknowledge our volunteer judges. We would like to welcome Li Young-Shen. She is the incoming chair of the Protein and Co-Products Division. Dr. Chen is a professor at the University of Alberta and a Canada Research Chair. She also serves as an associate editor of Sustainable Food Proteins, a journal of the American Oil Chemist Society, which just last month celebrated its first issue. So we're really excited about that. We also have Professor Vishnu Karki, a professor at South Dakota State University, Yongwei Li, a professor at Kansas State University and incoming treasurer of the Protein and Co-Products Division. And finally, Janitha Wanasandara, a senior research scientist with Agriculture and Agri-Foods Canada. She is the co-editor of the Sustainable Protein Source book, which is currently going through its second edition. Janitha is also a fellow of the American Oil Chemist Society. So as you can see, we have an expert panel of scientists with us today for judging. So this is how the ePoster pitch competition is going to go today. First, each student will present for up to 5 minutes. Second, after each student presents, there will be a 7-minute question and answer period. Our live streaming audience may post any questions or comments or congratulatory notes in the chat during the live stream. And we will try to address as many questions as we possibly can during the 7-minute limit. After all presentations have ended, we will pause the live stream for a few minutes to allow for final judge deliberations. During that time, the viewing audience will be able to vote for their favorite pitch. So then finally, we will announce the first, second place, and audience favorite winners. So make sure that you vote and make sure that you stay tuned to the end so you can find out if your choice was the winner. So that is, as long as that's clear with everyone, we will get started. It is time to begin. Our first presenter Our first presenter, Yifu Chu, yes, that was right. Yifu Chu is a graduate student at the University of Alberta. Yifu Chu will present on the topic of his ePoster, Softball-Bearing Lubrication of Size-Controlling Whey Protein Microgel for Designing Fat-Reduced Food Products. Yifu, take it away.
Thank you for the nice introduction. Hi, everyone. My name is Yifu, and I'm a PhD candidate in the University of Alberta, Canada. So today is my great pleasure to present my research, which is titled Soft Ball-Bearing Lubrication of Size-Controllable Whey Protein Microgels for Developing Fat-Reduced Food Products. So according to WHO, obesity has tripled over the past 40 years, and the contribution of calorie-dense food to this global crisis cannot be ignored. So designing fat replacer to mimic the function and the sensory properties of fat droplets can be a potential strategy to decrease calorie intake. So I believe most of you have tried various kinds of fat-reduced food, and you may find that they have inferior mouthfeels compared to their full-fat counterparts, such as creaminess and thickness, to be specific. That's because the fat droplets can provide texture and the mouthfeels to the food. So to mimic such sensory properties, we have developed protein microgels, and our overall hypothesis is protein microgels with similar shape and size to fat droplets with low calorie can potentially mimic the creaminess and the thickness perception of fat droplets. So in our lab, we first developed a faster method to prepare uniform and size-controllable whey protein microgels through a water emulsion, which has very strong industry interest. So what we did is just easily mix the whey protein solution and the alginate solution under pH 7 and heat for around 10 minutes. So during the heat treatment, the water emulsion will be formed spontaneously, mainly because of the electrostatic repulsion in between the two biopolymers, and at the same time, the microgels will be formed by gelation of the water emulsion droplets during heating. Because of the excellent gelling capacity of whey protein. So this method is more advantageous over other conventional strategies like microfluidic or emulsion template method because of the ultra low interfacial tension of the water-in-water interface, which means it doesn't need to add any surfactant or chemical modification or high energy input to control the size, which is suitable for food and biomaterial applications. So the microgels are quite spherical, which can potentially promote the ball-bearing lubrication effect to generate a creamy mouthfeel. So with this method, we can easily realize a process control of microgel size from 1 to 20 micrometers with a very uniform set distribution. If you check the set distribution data and the optical microscope here, and then you can also find the 3D cross-linked network in the ICM images. So when we talk about trying to mimic the texture-related attributes from fat droplets, we can try to measure the rheological and the tribological properties using a rheometer to predict the sensory properties. So for the rheological tests, these two figures show that the emulsion with 30% oil with 1.5% of microgels showed comparable elasticity and even higher viscosity to that of emulsion with 50% oil, but without microgels. Though the confocal image also showed that the microgels can work as fillers in the continuous phase of the emulsion system to improve the emulsion stability and can maintain the viscosity for over 1 year, which can potentially enhance the thickness perception of the food. Then what we got from the tribological test is a straight-back curve showing coefficient of friction as a function of sliding speed. So if you look at this figure, the microgel suspension showed a much more efficient decrease of friction with the increase of sliding speed, which represent a higher lubrication performance compared to the whey protein solution itself and even human saliva. That means adding microgels to food product can potentially increase the creaminess perception by decreasing the friction in between the tongue and the palate because of the soft ball-bearing effects from the soft and the spherical microgels. So at the last, we'll consider apply these microgels as fat replacer in some real food models, especially emulsion-based food, including liquid or semi-solid food and emulsion gel or solid food. And that's actually one of the projects ongoing in our lab. And we'll have our panelists to judge whether adding microgels can improve the creaminess and the thickness perception of the fat-reduced food. So we're targeting to use these microgels to mimic the sensory properties from fat in the food that can benefit the fat-reduced food development to provide more fat-reduced food options for healthier outcomes, such as controlling the world obesity epidemic. Thanks so much for the attention.
Thank you, Yifu. That was a very interesting talk about the formation of microgels and their physical chemical properties. So I will turn it over to the judges now. It's your time for feedback and questions. And for those in the audience, please add your questions and comments to the chat. Do any of the judges have any questions?
Yeah, hi, I'm Vishnu here. Hi, Yifu. Very nice presentation. If I may go first, I have a couple of questions on your research that you presented. It is very well presented, and I really like the study, the concept that you have there. My question is, you chose the whey proteins for your microgel formation. Is there any particular reason? And if you replace that whey protein with other proteins, do you expect the similar results, or what is your thought process?
Yeah, thanks for this wonderful question. The reason why we choose whey protein is because whey protein has very excellent gelling capacity, so it's a very good biopolymer to get things started. So in terms of whether we're going to replace with plant-based protein, that's actually a research ongoing in our lab. So we've already get started successfully make plant protein-based microgels using protein from plant resources, mainly from legume. And yes, if you ask me whether we can make microgels using plant-based protein, the answer is yes, and we are able to achieve that using just the same method as I mentioned in the presentation.
Okay, thank you.
Yeah, thank you for the question.
Okay, so I have a question. Yeah, Yifu, so very interesting study. So I saw you have the rheology properties of the protein gels. So did you compare like the sensory, like human sensory for the real fats with your protein gels? Really, if we are going to taste that protein gel, how would that have that similar texture as the real fats?
Are you asking like for the real sensory test, like compare the microgel to the real fat?
Sensory attributes of the gels compared to the real fats.
Yeah, unfortunately not yet. So that's actually the a project ongoing in our lab. So we are going to like compare the food like without fat, but with microgels and compared to their full fat counterparts. Yeah, but we don't have any results yet.
Sure, thank you.
Yeah, thank you.
Go ahead, Dr. Wanasan.
Yeah, Yifu, very interesting work and presentation. My question is related to your microgel. In microgel preparation, you used whey protein and an anionic polysaccharide, right?
Yeah.
So in your evaluation, you say the lubrication of this microgel is much superior than lubrication behavior of human saliva. Did you check with the anionic polysaccharide only how it's the lubrication?
So yeah, we do. We did test the lubrication effect of polysaccharide, which is alginate. So what we found is the alginate is not really effective that to decrease the friction with the increase of sliding speed. So what we got from that is microgels is much more efficient in terms of decreasing the friction in our tribological test, mainly because of the ball bearing effect, as mentioned in the presentation.
Okay, good.
Yeah, thank you for this question.
Just one quick question on the heat. What kind of temperature do you use for this microgel formation?
We use 90%, 90 degree.
90 degrees Celsius.
Yes, yeah.
Okay. So are these temperatures chosen based on the protein denaturation temperature? Are the basis of that?
Yeah, it's chosen because the temperature for for the gelation of the whey protein, and of course it's above the heat, the denaturation temperature.
Yeah, thank you.
Thank you for this question.
Yeah, another question. Improving emulsification properties, basically it's emulsion stabilization properties, right? Emulsion stabilization, right? That's what you observed.
Sorry. I'm sorry.
With the microgels, when you made emulsions, it is mostly emulsion stability. That is what you observed. Or is it how much oil can be emulsified? Which one did you observe?
Like for the water emulsion or the oil. So the water emulsion is kind of stable as we observed. Of course, the interfacial tension in the water interface is actually very low, so it should be stable during the microgel formation. I hope that can answer your question.
Yeah, because you mentioned it works as a filler than an emulsifier.
That's for the oil and water emulsion. So for the stability, for the stability of the oil and water emulsion, as the microgels can work as fillers, it can to some extent prevent the coalesce of the oil droplets. So during monitoring the phase separation, the stability. So the emulsion with microgels can maintain its stability without phase separation for over 1 year.
Okay, thanks.
Thank you for this question.
All right. Are there any other questions? I believe we are at time. So let me congratulate you, Yifu. That was a very good job that you did and a very interesting discussion. Thank you.
Yeah, thank you. Thank you, everyone. Thank you for the questions.
All right, our next presenter is Sumanjot Kaur, a graduate student at the University of Arkansas. Her presentation is on generating nanoporous protein aerogels from defatted rice bran via supercritical carbon dioxide. Sumanjot, the floor is yours.
Thank you for the nice introduction. So as we know, the demand for the plant-based protein and sustainable approaches has captured the great attention of food industries, leading to the great interest in processing bioproduct utilization to address economic and environmental concerns. So my today's talk will be focused on generating nanoporous protein aerogels from defatted rice bran by supercritical carbon dioxide technology. As we know, the rice is a major cereal food crop and Arkansas is a leading producer in the United States. A rice bran, a processing byproduct, corresponds to 8 to 10% of total grain weight, creating the disposable and economic problems. Although it is being used in the animal feed, but the food applications are still underutilized and restricted. The rice bran, which is a great source of the protein and has the high nutritional and functional properties. So we propose to convert the defatted rice bran into high-value aerogels for bioactive compound delivery using supercritical carbon dioxide drying. So what is supercritical carbon dioxide drying? Carbon dioxide, which is non-toxic and have very low critical values. When we heat beyond this critical point, like increase the pressure and temperature, it will exist in a fluid state and has the properties of both liquid and gas, for example, viscosity, diffusibility, and density. The gels dried using supercritical carbon dioxide drying are named as aerogels. These aerogels are biodegradable, have very low density, high surface area, and porosity. The aerogel formation includes 3 different processes: hydrogel formation, solvent exchange, and supercritical carbon dioxide drying. So in first step, we mix the water and biopolymer at different concentrations to form the hydrogel. Due to the high solubility of ethanol in supercritical carbon dioxide, we introduce the hydrogel in the different concentration of the ethanol solution to exchange the water with ethanol in the food matrix. Then the formed alkogels were dried using supercritical drying, which further preserved the porous structure and reduce the capillary stress on the food matrix and prevent the shrinkage. The drying parameters were the 10 megapascal pressure, 40 degrees centigrade temperature with the carbon dioxide flow rate of 1 liter per minute, and further characterized for textural and functional properties. So as we increase the concentration, the 15% concentration reported to maintain the better shape of the hydrogel. Below this concentration, it was difficult to maintain a shape, and beyond this concentration, it was becoming more denser hydrogel with high viscosity. So the SEM images further revealed the 3-dimensional open porous structure of the aerogel as compared to the extract. These findings were further justified using the high surface area of 34.5 meters square per gram and the low density of 0.19 gram per centimeter cube and the high porosity of approximately 87%. During aerogel formation, the gelation causes the deformation of crystalline peaks in the aerogels and the amide bonding deformation. This which resulted to the decrease in degree of crystallinity. As the degree of crystallinity decreased, it improved the thermal stability of protein aerogel and the stability of the aerogel, which also resulted in the high emulsifying and foaming capacities. So overall, this project helps to develop the nanoporous materials from the protein-based aerogels for bioactive compound delivery and develop the functional food ingredients, propose the new avenue for utilization of rice bran, and improve the sustainability in the rice production by reducing the waste generation. Thank you.
Thank you, Sumanjot, for that interesting talk about how we can apply, eventually get to applications for rice bran proteins. I'm going to hand it off to the judges. Does anybody have any questions or feedback for our speaker?
Maybe I can start. Yeah, so very interesting work. So one thing I want to clarify, did you use the rice bran protein extract or use the entire defatted rice bran for the aerogel preparation?
So we extracted different fractions for the starch and the protein and prepared the aerogel from the protein fraction. But our further study includes the aerogel formation from different protein fractions so that we can see the textural and functional properties of each protein fractions.
Okay, so just follow up on that question. So you have the protein extract, there's albumin, globulin, prolamin, and glutathione. So what's your opinion on, how these different proteins could contribute to the aerogel property? Would you prefer albumin may perform better than those, globulin, or, the prolamin even better? What's your thought on the, protein property and this aerogel property relationship?
So these all different protein fractions are have different solubilities. So one fraction is water soluble, alkaline soluble, ethanol soluble, and salt soluble. So I believe that globulin and albumin can form better gel formation, and in compare of this solubility, we can contribute in our further study. Maybe I can't reveal about their stability because each fraction has the different solubilities.
Maybe I can go the second. It's quite an interesting area to make aerogel from biopolymers, including protein or polysaccharide. I know there are some researchers in Canada, they are using supercritical technology to make aerogel from beta-glucan. So I just asked you how you compare your rice protein aerogel versus beta-glucan. What do you expect they will be different, how they will be used in different food applications?
So the rice bran protein has high functional properties, like it's a hypoallergic and gluten-free. So it has wide application in gluten-free products or the food for infants.
Then if I follow up, how would you think the beta-glucan aerogel can be used?
So which one is better?
Or they can be used in different fields, right?
Yeah. So yeah, this rice bran protein aerogel can be used for bioactive compound delivery, and most of the research is also been going on for the drug delivery processes too.
Mm-hmm. Okay. Maybe just one comment. If you protein you mentioned, you got a very good point. It's gluten free and it's byproducts. On the other hand, beta glucan is a dietary fiber, so they will have different space in food applications. Yeah, I will stop here.
Maybe I can go next. Again, congratulations, Sumanjot, for being here and then also having this great presentation. My question is along the line of Dr. Yonghui. So you use the extract that you mentioned, and then you have different fraction, but you use for this study the whole extract, I believe.
Yes.
So what percentage or what purity of the extract do you get in this process? Because I'm not very familiar with the rice bran protein extraction. What method do you use and what kind of purity of these proteins you get at the end of the process?
So it's very difficult to extract the protein from the rice bran because it has very complex structure, because protein is bound in the myelopectin cell walls. And we first extracted the protein based on the alkaline method. And we have not determined the purity of the protein, but we just measured the percentage yield we have obtained and compared to what was mentioned in the literature.
Okay. So right now, do you know the proportion of this albumin globulin in your extract, or you need to determine that?
So that study is still going on in our lab. So maybe in future, I will discuss that study too.
Okay. And so supercritical carbon dioxide is a technology that is it scalable for your process if you were to going in an industrial application that you mentioned in your last slide? So do you see there is a potential challenges, or is this technique already been applied in the industry? What is the current status?
So the problem with this technology till now is its high installation cost. Other than that, it is economically feasible and environmental friendly, and is a better way to utilize the byproducts from the processing industry to form the new functional food ingredients. So whenever we talk to rice industrial people, so every time they have one question about high installation cost.
Okay. All right. Thank you. Yeah.
Yeah, I have a clarification or question and a question, Sumanjot. It's a good presentation and interesting, the rice bran, improving the use of rice bran in different applications. So you mentioned about water solubility, right? Mm-hmm. So in terms of aerogel, have you, is high solubility good or low solubility good? Can you describe water solubility properties, how they can be used in aerogels, describing aerogels application?
Yes, so after forming the aerogels, overall crystallinity of the protein structure is being decreased, which is directly proportional, like more of the amorphous structure is being obtained. So that's why that was the reason why we are getting the higher solubilities, and higher solubilities are required when we utilize for bioactive compound delivery. So recently we are doing the in vitro digestion study, so that how it is going to be digestible in our human body. So the solubility is directly related to how the human body in the how the drug or the bioactive compound loaded in that aerogel have the accessibility in the human body.
Okay, so basically this is aerogel is the matrix that you want to deliver the compound. So
yes,
as it is breaking down just by dissolving in water,
yes,
high solubility is expected in your product.
Yeah.
Okay, good. Did you talk about starch at all?
Yes, no. So during our extraction, we extracted different fractions like starch, crude starch, and the proteins. So in this study, I have discussed the proteins, but we are also doing the similar study with the starch and crude starch, which is, I think, a great source for bioactive compound delivery too.
Right. Yeah, because I see it's only about 14% protein, total protein in the bran.
Yes.
So
yes.
a large fraction of dry matter is left out if you are just
yes.
looking at only 14% of the dry matter.
Yes.
Yeah, so to do the whole sustainability, to satisfy the sustainability equation,
yeah.
we have to utilize the other fraction, other
yes.
Valuable components of rice bran.
Yes,
as well.
We are also utilizing the starch and crude starch crude starch so that we can determine fiber content.
I hate to interrupt this interesting conversation, but our time is up and we have to move on to our next presenter. So thank you very much, Sumanjot.
Thank you.
And stay tuned for our next presenter. Our next presenter is Emerson Nolasco, a graduate student at the University of Nebraska-Lincoln. His presentation is on understanding the role of gastrointestinal microbiota in modulating the anti-hypertension effect of egg white hydrolysate. Emerson, the floor is yours.
Thank you for your introduction. In this presentation, we're going to go through how egg white proteins can help with hypertension. I'm going to start talking about cardiovascular diseases, which is one of the leading causes of mortality in the world. Such conditions include heart failure, in which hypertension is a risk factor. Risk factor is something that is associated with the increased risk of suffering a disease. Hypertension has several mechanisms, but for the purpose of this study, we're going to focus on 4: oxidative stress, vascular inflammation, the gut microbiome, and the renin-angiotensin system. There are already drugs that treat hypertension, but they remain inaccessible for low socioeconomic population. And also, they address only one of these mechanisms. A potential cheaper alternative that could address one or more mechanisms are food hydrolysates. Here we take a food protein and through processing techniques, we chop or hydrolyze the protein into small peptides. The peptides compose the whole hydrolysate, and once this hydrolysate is ingested and goes through our gastrointestinal system, it gives 2 fractions. A small fraction, which have shown to be more bioaccessible and bioavailable, meaning that it can be easily absorbed in our body, and also a digestion-resistant fraction, which there is not much information out there, but I'm going to talk about it more in my next slide. So more about food hydrolysates. In this study, we're using an egg white hydrolysate because it has shown to have be a good source of anti-hypertensive peptides. Studies have shown that up to 30% of uncooked egg white remains undigested. And here we hypothesize that this digestion-resistant peptide after the intestinal digestion can reach the colon. And there it encounters some microbes in our gut called gut microbiota. These microbes can have enzymes that cleave these digestion-resistant peptides and produce some metabolites. The metabolites can be detrimental to the host, but also they can produce positive metabolites such as short-chain fatty acids, which have shown to be anti-inflammatory and anti-hypertensive. So there remains the question, how will the gut microbiota respond to the digestion-resistant fraction of the egg white hydrolysate? Will it help in its antihypertensive activity? To answer this question, we prepare our own egg white hydrolysate, and we through enzymatic hydrolysis, and we subjected it to a simulated gastrointestinal digestion. After that, we fractionated with a molecular weight membrane separation, and the small fraction was used to evaluate its bioactivity in human endothelial and epithelial cells. The digestion-resistant fraction was used to evaluate how the gut microbiome responds to it. We observed that the small fraction was able to reduce oxidative stress in gastrointestinal epithelial cells at 2 concentrations. Moreover, the small fraction also reduced inflammation in endothelial cells by reducing the expression of an inflammation marker called VCAM1. This was done through Insel Western. Next, we took the digestion-resistant fraction and we fermented it with the gut microbiome of a healthy patient and compared it to a control such as peptone, and the fermentation was for several time periods. Here we found out that the egg white hydrolysate produced more positive metabolites such as propionate, which have shown to be anti-inflammatory and antihypertensive. And also, the egg white hydrolysate produced less negative metabolites compared to the peptone. Lastly, we evaluated the antihypertensive effect in spontaneously hypertensive rats by treating them with the egg white hydrolysate for 12 weeks. And at the end of the 12 weeks, we observed there was a significant difference in the mean arterial pressure, which was lower for the egg white hydrolysate treated group compared to its control. The relevance of this study relies in that it validates the food hydrolysate as an alternative to single target drugs, and also it lays the groundwork to develop new type of preventive alternatives to reduce hypertension progression. Thank you very much for your attention.
Thank you for describing the fate of egg whites in our digestive system, Emerson. With that, I will turn it over to our judges. And for anyone watching out there, don't forget that you can also contribute your questions to the chat. Judges.
Maybe I can get a start. That's very nice work, Emerson. I enjoy your presentation. I noticed that in your title, you mentioned their interaction in the presence of microbiota in the gut. But in the presentation itself, I don't see where's the role of microbiota in your processing. Could you give more explanation here?
Yes, definitely. Thank you for your question. So here the role of the gut microbiota relies on fermenting that fraction that was not digested in our small intestine. So once they ferment that, they produce these metabolites that can have beneficial effects. And one of those metabolites have been shown to be propionate. So when propionate is produced in our gut, it can have anti-inflammatory effects, and it can also show anti-hypertensive effects, which address two of the mechanisms of hypertension, which is vascular inflammation, as well as the oxidative stress, for example, as well.
Can I further ask you, those functions are normally known for polysaccharide or prebiotics. How could you explain the mechanisms why a peptide will have this effect?
Yes, that's a very good question as well, because most of the literature relies on or says that most of these short-chain fatty acids are produced by fermenting dietary fiber, but there are some studies as well that have shown that, for example, by fermenting peptides from soybean, they produce short-chain fatty acids such as butyrate. In our case, because we see propionate, we have seen some studies that have shown that there is a pathway in which the gut microbiome can ferment amino acids such as alanine and convert them to pyruvate. And then from pyruvate, they convert it to lactate, and from lactate to propionate. So some microorganisms can have these enzymes, and as well that enables them to produce the short-chain fatty acid through this pathway.
Can I ask the further questions regarding the literature? You mentioned that there's a literature that maybe inspire research that use soya protein that can trigger the activity of the microorganisms in the gut. But we know soya, it's a source of both protein and a dietary fiber. So in that literature, the researchers use the whole soya bean or protein isolate or concentrate.
Yes, I didn't explain it very clearly, but they use peptides only, isolated peptides from soybean protein.
Okay, thank you.
Thank you for your questions.
Maybe I can continue. So yeah, so for your egg protein hydrolysis, my understanding is you use Pepsi to prepare that hydrolysis, right?
Well, I cannot say the details of which enzymes we use because it's under patent, but we use an enzyme under acidic conditions and an enzyme under alkaline conditions. So this helps us to produce more of these anti-hypertensive peptides by exposing the aromatic amino acids in the N and C terminals of the peptides, which has been shown to be responsible for the antihypertensive activity.
Okay, yeah, I was asking because, after that preparation of the hydrolysis, and then you have the GI, stimulate GI digestion. So, if you just use the egg white protein directly without that pre-hydrolysis and then move to the GI digestion, would you have like, that some anti-hypertensive peptide as well, or did you compare this type of, different treatments?
Yes, so there has been studies done on the antihypertensive activity of the hydrolysate compared to the egg white. And it was shown that because of the enzymes that are used in the preparation of the hydrolysate before GI digestion, those peptides are still there after the simulated GI digestion. So they can be absorbed in our body and therefore exert their antihypertensive activity. While compared to the egg white, because the peptides that are produced are different and don't have this specific structure to enable them to exert antihypertensive peptide, they don't reduce the blood pressure at the same level of the hydrolysate.
Thank you.
Yeah, thanks for your question.
Hi, Emerson. Yeah, very nice presentation. I am curious, in making your egg white hydrolysate, so you said it's a different enzyme combination you used and you are not going to reveal that. Did you use the raw egg white or any heat treated egg white? Is there any enzyme inhibitor activity towards these enzymes in the substrate?
Yes, thank you for your question. We use egg white powder. We reconstituted up to a percentage of 2.5%. And then we perform a heat treatment of 80 degrees Celsius for 2 minutes to allow a partial denaturation of the protein so that the enzymes can cleave it more efficiently and therefore have more peptides.
Okay, I wish you explained that or mentioned that at least partially denatured egg white was used for hydrolysate preparation. Okay, good. Yeah, that's my question. Thanks.
Thank you.
Again, Emerson, very nice presentation. I think we are running out of the time. So very, very quick question. I think as Dr. Yunghoi suggested, I think the control, though there have been a study that you mentioned that there are without the hydrolysate, and I think your study should include a control. That's what in my opinion, without the hydrolysate and hydrolysate, and you compare, that would, I think, add a different perspective or maybe a control parameter in your study. And just a quick note on the microbiota. Do you know what kind of microbiome did you use when you had a secondary fermentation for the digester? Do you have a profile of those microbiomes?
We do not have the profile of the microbiome yet. That is ongoing work we're performing right now. We did extract the DNA and sequencing it, but we're still on the process of that data analysis. But yeah, if there's for example, one interesting thing is that there's some Clostridium propionicum, for example, from the Clostridia genus, have shown to be one of the responsibles for the production of propionate. So that could partially explain why we see this propionate increase. Thank you.
Wonderful. Thank you.
Thank you for provoking this interesting discussion, Emerson. Our final presenter is Ruoshi Xiao, a graduate student at Kansas State University. She will present on comparative analysis of the physicochemical characteristics of kafirin protein extracted from various grain sorghums and their distillers' grains. Ruoshi, take it away.
Thank you for your introduction. Hi, everyone. My name is Ruoshi Xiao. Today, I'm going to talk about the comparative analysis of the physicochemical characteristic of kafirin proteins extracted from various grain sorghums and their distilled grains. So firstly, what is sorghum? Sorghum is a globally significant cereal crop, and it can also be a potential source of innovative plant proteins. The protein content of sorghum varieties is around 6 to 18%, with an average of 11%. The primary storage protein in grain sorghum is called kafirin. In US, sorghum is mainly used for animal feed, biofuel, and alcohol production. Distillers grains, also called DGS, are the major co-product after ethanol fermentation. And there is a need for the value-added use of the DGS. There is a flow chart on the right side shows the fermentation process of the sorghum kernels. So in our study, there are 7 different kinds of sorghum kernels, including red, white, black, and waxy varieties were used. A portion of each type of sorghum were used to produce Chinese baijiu, which is kind of famous liquor in China, and the resulting DGS were collected. The original sorghum kernels and their DGS were grinded into fine flours, and the kafirin proteins were extracted using glacier acetic acid method. Then we analyzed the physicochemical properties and the functional properties of the kafirin proteins, and in comparison of some commercial plant protein, including soybean protein isolate, pea protein isolate, and wheat gluten. The objective of our experiment is to explore the effect of sorghum type and fermentation process on protein properties. So following are our results. The extracted sorghum had protein content range between 75 to 85%. And the SDS page result shows that the kafirin from different types of sorghum DGS had a similar profiles, while new bonds in the range of 15 to 20 kilodalton and 25 to 37 kilodalton were observed for the kafirin from the flowers. And the kafirin from black sorghums has slightly lower in vitro protein digestibility compared to that from other sorghum DGS and their flowers, which may be due to the much higher total tannin content. And the oil holding capacity values of the extracted protein ranged from 2 to 3.2 gram oil per gram protein. And the water holding capacity value of the extracted protein ranged from 2.1 to 3.4 gram water per gram protein. And this is the emulsification test. Black sorghum DGS protein had the highest emulsification capacity among all the kafirins, which is even similar to the commercial SPI. And the picture on the right side shows that the emulsion layers of all types of proteins. So in summary, we can find out that the protein composition and secondary structure generally remain similar for the kafirin extract from different types of sorghum or the DGS. The protein in vitro digestibility was negatively correlated with the total tannin content of the protein. The protein functional properties vary to some extent amongst different sorghum types and with fermentation. And now our research is ongoing to extract the entire sorghum storage proteins, including kafirin and glutenin, and explore their food application. So that's all of my presentation. Thank you for listening. And please feel free to ask me if there is any questions.
Thank you, Ruoshi, for informing us about this new source of protein and its physical properties. I will turn it over to the judges for questions and comments.
Ruoshi, I have a question in your presentation. It's very interesting. Sorghum is a crop that is known for high tannin, and you are showing some varieties that have low tannin. And I noticed in your presentation for in vitro digestibility, the variety white 4325 has lower digestibility, although it has 0 or no tannin in your bar graph. Can you explain that in relation to your conclusion that tannin and in vitro digestibility relationship with negative 0.42?
Oh, that's a really good question. As we know that tannin is a kind of like anti-nutrient compound. When we test the digestibility, the tannin will react with the protein, and we will bind the protein, and to reduce the protein digestibility in this and I think it's because that protein will like bind to the tannin, and to reduce the protein reaction rate of the protein reaction with the enzyme. So at that time it will reduce the digestibility, and in the other side the tannin will also affect with the enzyme we use to test the digest. So it will affect the enzyme efficiency of that. So that's maybe the reason that why the protein digestibility is really low when the total tannin content is high.
Yeah, that is true for the high tannin one, but I'm particularly asking the white variety. I believe it has no tannin, and that's your data also showed it has no tannin, but still the digestibility value is low.
I think that may be because that the sorghum types of that, the like natural structure of this kind of the white sorghum, so it will to make it have a lower digestibility, not only the result of the tannin.
Okay, so in your conclusion, you show a very weak relationship, but it is actually type of protein is the one in question rather than relationship with tannin, isn't it? Because 0 tannin also has low digestibility compared to the high tannin.
Yeah, I think this part of it will have like, we should have more like experiment to test what the other effect to affect the protein digestibility of that.
Right. Yeah, sure. Thanks.
Hi, Rosie. Very nice presentation. Just to follow up on Janita's question. So as you have two characteristics, that is looking at the digestibility and Janita's last remark was probably the protein has to do something to with the digestibility possibly. Have you characterized these proteins because you have included different varieties of the sorghum, and then you extracted the kafirin proteins? Is there any other than the STS phase, because that shows almost have similar characteristics? Is there any other properties you could measure or characterize the proteins in other way, look at the amino acid profile? Is there any way to do it? Just asking your thought.
I think
what would you do? What would you do to identify, basically to simplify the question for you? To look into the protein aspect of the kafirin, right? Kafirin, yeah.
Yeah. So what your question is to like, it means that what other tasks we can do to like
what would you do next to identify what is the different, how the protein is different in X variety versus Y variety that you have, right? So is there anything you can do?
Yeah, I think we can I think we can do more tests about to find out the more like the fermentation process, the effect of the fermentation process of the protein. It's kind of like, for example, we can test more about the protein fraction to see there is any change about the kafirin, prolamine or glutamine about that, or we can test more like the free sulfate bind, and if there is any like chemical bind or some changes of that during the fermentation process.
Okay, good, thank you.
Thank you.
Okay, I have some question. You use FTIR as a tool to analyze the protein secondary structure in your study.
Yes.
And based on your conclusion that the protein in the sorghum versus the protein from the distilled grain, they have quite a similar secondary structure component.
Yeah.
I'm a little bit curious because I know in the baijiu making, that's typically maybe more similar to the beer making. We have the temperature treatment or some other treatment. I'm wondering, curious, why the proteins still maintain their secondary structure after those processing?
Yeah, that's really good questions. Actually, when I gathered that result, I'm really like surprised that there is really similar result. And what I thought is that they will have some like differences between that, because the fermentation process have the temperature and it will be some change, but the result shows that it's just similar, and even similar from this like different types all shows that there's no significant differences. I think there will be more lab tests we have to conduct to find out if there is any other change on our protein.
Can I further ask the question? You know, I know FTR is just the one tool to test the protein conformation. You know, based on FTR analysis, maybe the secondary structure don't have any significant change. Would you consider, when you have progress of your research, maybe could you suggest another tool or another type of tool that will allow you to just to confirm the conformation change or not by different varieties or different processing?
I think we can like conduct tests about the protein protein fraction test to we can use the HPLC SEC HPLC to see that the if like kind of protein fraction change, like if when we before the fermentation process, we will have like this like maybe 30% pronamine and 20% glutamine, and after fermentation process we will have like more like 40% 50% prolamine and the 10% glutamine. They will like, this is maybe we can find out the change into the different kind of the protein in the different type of protein in the like sorghum protein. I think this is a test we can like conduct more.
That's very good. Yeah, that's very good, because I see you try to understand the protein conformation at each step of the processing. That's very good. Maybe suggestion, maybe you can consider fluorescence spectrometer or circular dichoisms. They can analyze the protein tertiary structures. Maybe tertiary structure is open, but the secondary structure is still maintained. But thank you so much to you know to have a more further explanation of the experimental design.
Thank you so much for your suggestion.
Unfortunately, we've run out of time. I hate to interrupt this interesting discussion, but we need to give our judges time to deliberate on who is first and second place. And then I also want to remind our audience that you are also welcome to vote, and the polls are open for that. So excuse us while we deliberate. And I just want to thank all the presenters. This has really been a very good event and lots of great discussion here. All right, so all of the votes are in. I wish I could do a drum roll, but the first place winner is Sumanjot. Yay, And second place is
you.
Emerson. And the People's Choice Award is also Emerson. So congratulations to everyone. Everyone involved was really impressed by the presentations and the diversity of the science. And we all, agree that we learned a lot. And as you could tell, we had a hard time shutting down Q&A because we all really enjoyed talking to you. So thank you so much for all the competition presenters and judges and the AOCS Foundation for funding the competition, and to our audience for voting. We hope to see you all at the annual meeting.